Welcome, everyone. My name is Shawn Baker, and I'm the CSO and founder of AllSeq. And today I'm going to be talking to you about the impact of the $1,000 genome on medicine. So before I get into it, I just want to remind everyone that in the left-hand corner, there's a little Q&A button. So as I give the talk, if you have any questions or comments, please open that up and just ask anything you like. And at the end, I'd be happy to try to answer it. Okay. So with that, let's go ahead and get started. But before I actually get into the $1,000 genome and what it means for you, I need to kind of explain who AllSeq is. So we're the sequencing marketplace. What we do is we offer a free resource that matches researchers with sequencing providers. So if someone needs to do some sequencing and they don't know who to get it from, we help them find the right providers. Now, even though we do this, we actually don't offer any sequencing ourselves. We don't own any of the sequencers. So that means we're able to maintain a neutral stance. We don't have a vested interest in any one of the technologies. So without any overt bias, we can guide people to the right thing, right technology, the right provider for their particular research project. Doing this has really given us a unique perspective on this market. Because our users trust us with this neutral stance, they let us know what their budgets are. We know what kind of projects they're running, what kind of species they're running, what technology platforms they want. We also see exactly how much their projects cost. So this gives us a really interesting and unique view into the market, and it's part of that I'm going to share with you today. So the big news this year, of course, is that the $1,000 genome is finally here. Sort of. There's actually some caveats. I'm going to walk you through those caveats. So what's caused all this? This is from the announcement earlier this year from Illumina, with the launch of their HiSeq X Ten. This is their latest instrument, their largest and the highest output. So what exactly is this instrument and how is it getting us to the $1,000 genome? So it's based on their, at least loosely based on their HiSeq 2500, their current flagship product, but it has a greater output. It's actually able to generate 600 gigabases of data per day. And over a 3-day run, that means it can output 1.8 terabases. That's 1.8 trillion bases, incredible amount of output. That's actually enough data that that single instrument can generate 5 human whole genome sequences per day. And then of course, if you run it all year, that means that one instrument can generate over 1,800 genomes per year. So how does it do this? What are the major advancements? 2 things. One is a new flow cell. This is a pattern flow cell. It allows the instrument to pack in more data per run. And then the other thing is improved optics. And what these optics do is allow that run to be shorter. So between these two, they're really able to jack up the output per day per run. So that's giving you all that data. And then, of course, they've reduced the price to match. So this sounds really great. New instrument, lots of output, better than anything that's out there. What's the catch? What's going on here? So the advent of next-gen sequencing and as it's gained popularity has really broadened out its usage. People are basically using it for anything that has DNA. Anything has DNA, it's being sequenced. Just the amount of applications, the amount of species that are being looked at is just tremendous. And we see everything on AllSeq Sequencing Marketplace. The HiSeq X Ten, however, is only able to look at one species, and that's human. Not only that, it's only able to look at humans using one application, and that's whole genome sequencing. So that means you obviously can't look at other species, can't look at cattle, can't look at plants, only humans, but also you can't look at other applications. You can't use exome sequencing, you can't use it to look at, say, for example, the methylome or the transcriptome. Just that one application and one species. But it is able to deliver the $1,000 genome. So I want to go ahead and walk you through exactly how that happens, what it's able to do. So the first thing is if you want this $1,000 genome, you've got to buy an instrument. And this instrument, the X10, is the most expensive instrument on the market at $1 million. So that's already pretty pricey. But Illumina throws another wrinkle in there. You're not allowed to buy just one. You actually have to buy 10 of them at a time at least. So that means you have a minimum capital outlay of $10 million. It's already a pretty big barrier for most people. So how does the actual $1,000 genome break down? What is that money covering? About $800 of that is for the actual sequencing reagents, generating the sequence. 135 is for the instrument amortization, basically paying off that instrument one genome at a time. And the remaining $65 covers kind of everything else. So that means the library prep, both reagents and the labor for it, and then basic data analysis. But there are some things it doesn't cover, more specialized analysis, the cost of energy, the storage of the data, overhead, the building that it's in, all those kind of things, which are kind of hard to calculate globally. But if you have your own building, if someone else is paying the electricity and you buy this instrument, you can get the genome for $1,000. So let's walk through what that would actually mean if you're operating this machine. So I mentioned before that it's able to generate enough data for 5 genomes a day. So that means every day that you're operating it, it's going to cost you $5,000 to run. And of course, if you run it for a whole year, like you should, it's going to cost you $1.8 million to run, generating about 1,800 genomes. But if you remember, I said you're not allowed to buy just one instrument. You have to buy 10 of them. So operating all 10 instruments for a year is actually going to cost you $18 million, generating 18,000 genomes. But one more wrinkle, in order to get that instrument price down to $135 per sample, Illumina has calculated that you actually have to run this instrument for 4 years. So to get that $1,000 genome, you're actually going to have to spend $72 million. That's a pretty big capital outlay, and some people have been willing to do it. They've actually had 13 customers pony up that money so far. If you're not one of them, if you don't have the $72 million to spend, but you still want to get that inexpensive genome, all is not lost. That's one of the reasons we created the sequencing marketplace. You can go to allseq.com/$1,000 genome, and we'll help you get set up. We'll help you describe your project and get you mapped for one of the providers who actually owns one of the X10 instruments. Okay, enough about us. So with the advent of this X10 and bringing the cost of whole genome sequencing down so far, it's really sparked a renewed interest in the debate between whole genome sequencing versus exome sequencing. So I just kind of want to walk through that briefly to tell you what the differences are and why you might choose one over the other. So here we're just looking at a representation of the human genome. We have obviously all the chromosomes out here. If we zoom in, we're looking at a gene, and in this gene we have the exons, which are the protein coding portions, and then the introns, which are non-coding. And then of course, if you zoom in even more, you'll get the individual bases. So for whole genome sequencing, of course, as the name implies, it attempts to sequence the entire genome. So that's why I've highlighted all of these chromosomes, every region of them. In practice, it doesn't reach 100%. There are some regions which are just technically challenging, high GC content, a lot of repeats, things like the telomeres and centromeres. They're pretty hard to cover. But in general, the application covers around 95 to maybe 98% of the genome. For each gene, it tries to cover the entire gene. So again, both the exons and the introns, and then of course, every single individual base. Exome sequencing, in contrast, is much more focused. So it's still looking at the entire genome, but instead it's just focusing on individual genes. Also within those genes, it's really just focusing on the exons, or that protein coding region. And of course, within the exon, it's trying to get every single base. So we do see that it's still generating a lot of data, just not as much. In general, depending on the product that you use to do the enrichment, it's somewhere between 40 to maybe 60 megabases, representing 1.3 up to maybe around 2% of the genome. And that would be compared again to 95 to 98% of the genome. So given these two applications and the strengths and weaknesses, which one's better? It really depends on what you're going for. So let's just kind of walk through some of the characteristics of these. First, you have price. That's where the exome sequencing has traditionally been the leader. Up until recently, whole genome sequencing was anywhere from 5 to 10 times more expensive as the exome. So people with limited budgets or who wanted to sequence more samples tended to use the exome sequencing. With the advent of the X10, that difference in price is really diminished. It's closer to 2 times more expensive than whole genome sequencing now. So not nearly the price difference that it's been in the past. In terms of coverage, whole genome sequencing obviously has exome beat. Like I said, it covers up to 98% of the genome, whereas the exome maxes out at around 2%. So if you want to look at non-coding regions, whole genome sequencing is definitely the way to go. Another way that whole genome sequencing is actually superior is maybe a slightly surprising way, and that's in the uniformity. Due to the way that the exome pull down occurs, it actually leads to some non-uniform sequencing. Some regions are basically easier to enrich than others, and so you get some sequences that you just get a lot of data for, and other regions that you get much less data for. So overall, you actually have to run exome sequencing at a higher depth, say around 100x versus more typical for whole genome sequencing, 30x. And that's really just to make sure that you have minimum coverage in all the regions that you're interested in. Another area that exome actually shines, however, compared to whole genome, is on the analysis side. And that's just because you're looking at less data. When you're only analyzing 2% of the genome, and that 2% is actually the portion that is much better understood, it's just easier analysis. So some people who feel they don't have the time or the money or the expertise to look at non-coding regions will just concentrate on the exome. Now, some proponents of whole genome sequencing kind of agree with that, but they still want the better uniformity and the greater coverage of whole genome sequencing. So they sequence everything, but then they might restrict their analysis just to the protein coding portions of the exome. So they generate more data than they need, but then keep the analysis simple. They just look at a restricted portion. Okay, so this is all really interesting, lots of great developments. The question then is, what, if anything, is this doing for the clinic? And in order to understand that, it's important to look at the characteristics of what researchers want versus what clinicians want. So researchers traditionally they just want increased output. They've got large projects and they just want to generate more and more data, and that's what they've really been pushing the sequencing vendors to do. Of course, they also want to have lower cost, especially lower cost per base, so that each genome they sequence costs less and less money. And that's been a huge driver of this market over the past 5 to 10 years. They're also willing to tolerate, and in some cases demanding, of rapid updates. So once they buy the instrument, they know that's not the end of the equation. They know that company is going to keep developing new instruments, keep working to improve things, and they want those improvements to show up in their instruments that they've already had installed. So you'll often see updates come out in the field, sometimes twice a year, sometimes even once a quarter or once a month. These updates are sometimes about hardware, sometimes about software, but the whole point is to increase that output and increase, or rather continue to lower the cost for them. Clinicians, however, have a different set of needs. What they really want above everything else is simple ease of use. They really just want to be able to push a button and get an answer out. They generally don't care too much about the technology, and they just aren't as sensitive to these other things that researchers are. They also really need a quick turnaround time. So while researchers might be willing to wait weeks or even months for their large project and then analyze it for months and even years after that, the clinician is generally working with a patient sample and something for which they need an answer right away, more like days or even hours, because it's directly going to lead to some sort of treatment decision for that customer or that patient. The other thing that they really want that's in high contrast to researchers is they want stability. They don't want those rapid updates. Once they buy a machine and they get used to it and they see that it works, they want to make sure that what it did today is the same thing it's going to do tomorrow or next week, next month, or even next year. So they really value that stability. So how is this technology being actually adopted into the clinic? So there's actually only one instrument that is officially sanctioned for clinical research, and that's the HiSeq Dx. Last November, it was the first instrument to become cleared by the FDA. This has been so successful, however, that Illumina has decided to also submit their larger instrument, the HiSeq 2500, with a novel assay. And then Illumina's biggest customer, Thermo Fisher, is going to submit their instrument, PGM, for FDA clearance sometime later this year. We're still kind of waiting for some updates on that. So this is the only official instrument that's been cleared. It doesn't mean it's the only instrument that's actually being used in a clinical setting. Labs that are CLIA certified can and have been using other instruments. That may be changing. FDA's come out with some recent pronouncements. They're going to start looking more carefully at these LDTs, laboratory designed tests. So we'll see how that changes in the future. But for right now, people are using non-FDA cleared instruments in a clinical setting as long as it's a CLIA approved laboratory. So what kind of tests are actually being used right now with patient data? We've talked about whole genome sequencing and exome sequencing, but there are also smaller tests, so larger than a single gene, but smaller than exome, where people are looking at a large number of genes. So one example is Foundation Health. They have a test that looks at over 300 genes and genomic regions that are related to cancer. Another similar kind of thing is a company called Invitae. They have taken over 250 individual genetic tests and combined them into a single assay that they're selling for $1,500. This is kind of a unique business model for them. They say to the physicians, go ahead and look at all these 250 tests, decide how many you want to run, whether it's a single test, just a handful, or all 250. The price is actually the same, still $1,500. Not only that, the physician can order a single test or just a handful, look at the analysis, and if they don't see what they were looking for, if they do it quickly enough, they can actually go back to the company and get them to reanalyze the rest of those tests, because they're actually running all of them at once. When you choose just one or a handful, really what you're saying is just share with me that amount of data. So it's pretty flexible and relatively cheap, especially when compared to what those 250 individual tests would have cost. So that's an example. Those two are examples of more focused tests, but it's also being used, next-gen sequencing is also being used on the whole genome scale. Primarily, for things like cancer. That was one of the first things that was adopted for it. To be honest, this is often used as a last case scenario when nothing else has worked, either when the traditional genetic tests haven't shown something useful, or maybe when the patient isn't responding to the traditional drugs. A physician can order a whole genome test to look at all of the genome to see if there's something else they might have missed, something unusual that's going on that might indicate a different drug that they wouldn't have otherwise chosen. To be honest, it's clinical in that it's directly treating a patient, but it's really more of a research project. Once they generate all that data, the individual physician usually is not in a position to really look at it. They need to assemble a research team around them to quickly dig into it, people who are experts in genomics and in cancer to try to figure out what's going on. A similar kind of story goes on with rare childhood diseases. Same kind of thing. This is usually a test of last resort. They've either looked at other genetic tests and found nothing, or they just don't even really know what to look for. They'll sequence the child and often sequence both the parents, looking for what's different, what's going on, just any clue that they can get to indicate a potential treatment. But again, this is really more of a research project than it is a traditional clinical test, and just that it's not scalable. It takes a lot of people and a lot of expertise, a lot of time, and quite a bit of money, to be honest. A much more broadly available technology is a relatively new one, the non-invasive prenatal testing, or NIPT. This is an assay that if you'd asked pretty much anyone 3 years ago, what's the first whole genome sequencing assay to take off, probably no one would have chosen this. But it's incredibly popular. It's attracted a lot of commercial interest. I've shown to some of the companies here who are offering NIPT testing. And if you don't know what this is, it's basically a replacement for amniocentesis. So when pregnant women who are looking to see if there might be some disease they need to worry about in their unborn child, like Down syndrome, they would use the very invasive process of amniocentesis. It's now been replaced with a simple blood draw from the mother. Basically, there's enough fetal DNA floating around in the mother's bloodstream that they can actually determine these genetic diseases prior to birth. So it's much easier and much safer than the traditional methods. So with the impact that next-gen sequencing is having on research, and as it's moving into the clinic, it's important to ask, what's the impact on patients? What's actually happening to the frontline people here? And a big question that people are still trying to answer is, what exactly should these patients be allowed to see? So often a test will be ordered looking for a particular genomic region. They have a particular question in mind. But because of the nature of the technology, it's actually really easy to find things in other parts of the genome that you weren't really interested necessarily in asking about, but you might find something that you weren't expecting to see. This is often called the incidentalome. So harking back to the NIPT testing that we just talked about is actually a great example. So the test is designed to look at the fetal DNA to see if they have these rare diseases, but the fetal DNA in a mother's blood only represents about maybe 2 to 10% at the most of the DNA that's floating around. The rest is, of course, the maternal DNA. So even though they're looking for the fetal DNA, they're mostly finding out information about the mother. Mostly they ignore it, but occasionally they find something that's quite dramatic. Either a gene that shows they're extremely susceptible to cancer, or maybe something like Huntington's disease, where they know with 100% certainty that that mother's going to get it. And they then are put in a quandary. The patient came to us asking about their fetus, but we found out something about the mother. What should we do? And they're really looking for some guidance there. ACMG came out with some recommendations last year to try to help answer this. They came up with a list of 56 genes for which they said if there are abnormal results, they should be reported to that patient regardless of what the patient wished. Now, that actually was fairly controversial, and they got a lot of feedback from their users and from the general public. And so they've come back later this year and they've modified that. So it's no longer regardless of what the patient wishes. Instead, what they're saying now is when that test is prescribed, but before it's given, they should be able to opt out of any future results that they actually just don't want to hear about. So it's a discussion that goes on between the patient and the doctor to figure out what exactly do you want to know if we happen to find something. But this is still an evolving thing, a moving target, and everyone's really just working together trying to figure out what the best thing to do is. Okay, regardless of what you do, a big portion of next-gen sequencing actually is all about analyzing the data, and this is really critical. For clinical aspects, there's a lot of things involved, but the things that are most important are the tertiary types of analysis. This is annotating the genome and visualizing the data. Perhaps even more important than that is actually the reporting of the data. Physicians really want to see not so much the underlying data, but what does it mean? What should they do for their patient? So this is an example of a FASTQ file. This is essentially raw sequencing data. This is what it looks like. We're seeing, of course, the individual bases, and then we're seeing essentially quality calls, how good that data is. This is a very tiny example, just showing 25 bases per run and just 3 individual sequences. If you were to run this today, this would be a file, basically a text file that is 1 billion lines long. Obviously not something that a physician or even a researcher can easily just look through. Instead, what the clinicians are really looking for is something way down the line. They want this not just to be data, but they want interpretation. They want a medical report, something that's actually specific to their particular patient, that has the context of what the patient phenotype is, what the genomic anomalies are in that patient that are probably relevant to that phenotype, and even farther, they want to know what are some recommended courses that I should be looking at to help this patient. What are the drugs out there? What are the treatments that they need based on all this information? That's really what they want to get to. There's a number of companies working on this. It's obviously a hot area. We see new companies coming in all the time, trying to come up with really slick solutions. I would say nobody has really dominated this yet, but it's still such a strong need that it's pretty clear this is going to be a hot field for a while. So one of the advantages of running the sequencing marketplace is we get to keep a really close eye on the future trends. One of the things that a lot of people have been talking about is nanopore sequencing. This is actually using nanopores to measure the DNA as they pass through the holes, basically looking at how the DNA molecules disturb the electrical conductance. It's still early days. There is a company, Oxford Nanopore, who has an instrument out in early access, and I'm showing it here. It's called the MinION. It is essentially a handheld little machine, and it plugs in straight to your computer with a USB. So you just pop in some DNA, plug it into your computer, and it starts generating a sequence. It's still early. It's unclear how good it really is, but it is producing really long reads, which is kind of a new thing in this field. Unlike that $1 million HiSeq X that some people are buying to get the $1,000 genome, this instrument is probably going to cost more like $1,000. It obviously won't generate nearly as much DNA, but it's a really low capital outlay, so people are pretty excited about it. Another area that we see becoming more and more important, and we see people asking for this all the time on the sequencing marketplace, is single-cell sequencing. So for a typical sample prep, people are often looking at, tens of thousands to hundreds of thousands of cells all together. And so they combine all that DNA together and sequence it. So obviously the sequencing they're doing is generating an average across all of those cells. For many applications, that's not a problem at all. So if you're just looking at a standard genome, with very rare exceptions, it really doesn't change from cell to cell in your body, so it doesn't matter if you're looking at an average. But there are some applications where it could be critically important. So for example, in tumor tissues, that's going to be a mixture of a lot of different cell types, both tumor and normal, and even within the tumor, you'll have different states of mutation in the different cells. So if you could, it would actually be better to look at individual cells, look at their genomes one at a time, and then analyze them in aggregate, but being able to split them all out. So I'm just showing you here an instrument called the C1 from a company that's Fluidigm. They're kind of the forefront of the commercial instruments looking at single cells. The other thing that we're seeing that's of extreme interest in people is cell-free DNA sequencing. So the NIPT testing that I talked about before, that was an example. But another example that's not as well developed, but there's an incredible amount of interest in, would be cell-free DNA sequencing for cancer. Obviously, if you could detect DNA from cancer cells as they're just floating around in the bloodstream, well before you would see a tumor, that's going to lead to some really powerful, really early diagnostics. So we have a lot of people who are working on that. So just a reminder, everything I've been talking about today has been DNA sequencing, but that's really just the tip of the iceberg. So obviously there's a lot more going on. There's a lot of regulation of the genome, methylation, acetylation, things like that. There's also the RNA, so mRNAs that code for proteins, small RNAs, other non-coding RNAs. All of this stuff is of tremendous interest in the research field, and it's very likely to make its way into the clinic as well. It's just not there quite as quickly as the DNA side, but rest assured, it will continue to become more and more important in the future. So with that, I'll go ahead and stop. I want to thank you all for attending today. As I mentioned before, over in the left-hand corner, we have a little Q&A box, and I see some people have put some questions in there, and that's great. I'm going to try to answer some of those right now. So if you have some questions right now, please go ahead and type them in. If for some reason you don't think of the question today, or you just want to get some more information, please check out our website, allseq.com. You can also email me directly at shawn@allseq.com, and I'd be happy to help you out. So with that, let's go ahead and I'm going to open up the Q&A box and see what we have. So we have a question about coverage. What would be the recommended coverage to find both germline mutations and somatic mutations? That's a great question. We get questions about coverage all the time. How much sequencing do I need? Obviously, it affects cost of the project. And it really depends on what you're looking at. So if you've got a pure sample and you're doing something like whole genome sequencing that has pretty uniform coverage, the typical answer is around 30x coverage, sometimes up to 40x or 50x. So you're talking about 100 gigabases for a human genome. However, samples aren't always pure, and people aren't always using assays that have uniform coverage. So if you're doing something like exome sequencing, it's typical that they will go to a higher coverage, more like 100x. And again, that's really just to ensure that all areas that you're targeting have at least about a 10x coverage, because that's really what you need in order to be able to make sure you can tell the difference between a heterozygous mutation and a homozygous mutation. Now, the other area is when you don't have a homogeneous sample. You've got, say, that tumor tissue that might be a mixture of a bunch of different cell types, and you can't do single cell sequencing yet. In that case, you know, we've seen people go as high as 1,000x using exome sequencing. And that's because not only do they want the higher coverage to make sure they get that uniformity, but also if they're only looking at, say, one cell of interest out of 10, you know, 10% of the cells in that tissue are really what they want to look at, you need to have higher coverage. Okay, this is great. Someone else is asking about whether there are any indications that Illumina is going to extend the capabilities of that HiSeq X Ten. As I mentioned, it's only useful for humans and only useful for whole genome sequencing. You know, in both cases, those restrictions are purely marketing decisions. There's really nothing technical that would prevent it. When you buy the machine, you sign a contract saying that you won't do otherwise, and Illumina keeps, I think, pretty close look at that. But like I said, there's nothing technical to prevent it. So it's very likely in the future at some point that they will open it up, probably first to exome sequencing for humans is my guess. And then after that, you know, maybe to other things like methylation and transcriptome work. The reason they've done that really is for marketing. They already dominate the market. They own probably about 80% of the next-gen sequencing market. And rather than collapse that with just reducing the prices, what they wanted to do was expand the market. So they created this machine and created really low pricing so that people could start doing projects that involve not just hundreds of samples, but tens of thousands to hundreds of thousands of samples. This is when you're sequencing entire populations. And it's really until that market seems to really be well established, they'll probably maintain those restrictions. That's my guess. Another question that we have is talking about the actual price of a $1,000 genome. You know, it's called a $1,000 genome. It's kind of a shorthand, but as I mentioned, you actually don't get that price unless you have $72 million to spend over 4 years. More typical for people is to go through a provider to get that, and there the price is going to be obviously more than $1,000 because there's more things that go into that. There's some labor and some overhead and some profit that has to be made. In general, it's really going to depend on if you want anything unusual. You know, the more plain vanilla, if you're just using a blood sample and you don't need any really special data analysis, and you have very high volumes, say 1,000 samples or more, you can start getting that price down to somewhere, say, between $1,500 and $1,600. If you have a much smaller volume of samples you're working with, say closer to 10, it's going to be more expensive. Still, it's probably going to be somewhere in the $2,000 range. So it's still a pretty low price. Okay, with that, I think looks like we've answered most of the questions. So what I'd like to do is just go ahead and thank everyone for participating today and thank everyone who asked any questions. And then, of course, if you have any other questions, you think of anything else, please feel free to check out our website and or email me directly. I'd be happy to answer them best I could. Again, thanks everyone for joining. Appreciate it.